Greetings, everyone. Thank you for joining us in today's Power Talk with us here. I hope you are all doing well wherever you are. We will begin our Power Talk shortly. Also, it's already 1 p.m. and Professor Vimal Karani also has joined us today. So welcome again to one of the I3L Power Talk event series. I'm Irena and I will be moderating the talk today. So like what you have seen in the announcement, today we are having Professor Vimal Karani from University of Reading, United Kingdom, that will be sharing one of his projects about gene-nutrient interaction, or called GeNuIne. Hopefully I pronounced that correctly. So before I further introduce him, let me read our agenda and code and conduct for today. So we'll have like opening and forwards right now. And then Professor Vimal will be sharing his research. He will give a talk for an hour, and then we will continue with a discussion for around 25 minutes. And then we will have closing and also evaluation. And there are some rules to make our meeting today is going smoothly. The rules are that you are prohibited to activate the audio during the Power Talk session. But if you have any questions, if you want to ask specific questions during the talk, feel free to use the chat feature to ask questions. So yeah, you can ask the chat feature in this meeting room. And then the evaluation surveys will be distributed to the Power Talk attendees at the end of this event. This oral survey will be also used to record your attendance, your presence, and for getting the e-certificate, I mean. Right. Without further ado, I would like to introduce you Professor Vimal Karani. Hello, Professor Vimal, how are you?
Good afternoon. Yeah, I'm doing well. Thank you very much. Hope you're all well.
Yeah. So Professor Vimal is a professor in Nutrigenetics and Nutrigenomics at University of Reading, United Kingdom. He is also a deputy director, Institute of Food, Nutrition and Health. And he has academic background in molecular genetics and also bioinformatics, as well as biomedical genetics, and also certificate in academic practices. As we can guess from his title, he has done a lot of research and has published many papers on nutrigenetics and nutrigenomics field. One of them is GeNuIne, or gene-nutrient interaction studies, which made him rewarded the prestigious UK Nutrition Society's Silver Medal Award in 2020 for his contribution to the world of global nutrition. So I'd like to welcome Professor Vimal to give a talk on this topic. And Professor Vimal, I'll give the screen to you anytime you're ready.
Thank you so much for a nice introduction. So without any further delay, I'll share my screen. Just 1 second. Okay. Are you able to see the slides?
Not yet.
Because I was prevented from sharing the slide.
Oh.
Yeah.
Would you let me see?
Well, I'll try doing it again. Can you see the slides now?
Yes, we can see it.
Let me do the slideshow.
Okay.
You can see it now, right?
Yes, we can see it clearly.
Okay, perfect. Okay, a very good afternoon to everyone. Thank you all so much for joining the session, and thank you so much for inviting me to deliver this power talk to you all. So today I'm going to talk to you all about how nutrigenetics findings could be translated to precision nutrition. So I'm going to share some interesting findings from the GeNuIne collaboration and with regards to nutrigenetics and nutrigenomics research activities that I'm doing as part of the GeNuIne collaboration. So before I go into some of the interesting findings, I'm going to give an overview about some basics of genetics. So one of the most important things that you will have to know as part of nutrigenetics is the genetic heterogeneity. As the name implicates, heterogeneity means difference, and genetic is nothing but your genetic makeup. So your genetic makeup is so different from other people. So your DNA is unique for you and my DNA is unique for me. And that is what makes you and me different from each other. And that is the underlying basis for doing the nutrigenetics and nutrigenomic researches. And this concept of genetic heterogeneity, so that is the underlying basis why we want to go for a precision nutrition approach. Because we all carry a very unique DNA that is making us all very unique in different ways. The way I eat, the way I talk, the way I smile, the way I walk, all my behavioral activities, my personality traits, everything is being driven by your genetic makeup. So your genetic makeup is what you call as your genetic representation, and that is what you call as a genotype. Your genotype is a genetic representation, and what you see on the screen, some people are laughing, some people are talking, some people are giving different expressions. These are all the phenotype. The phenotype is nothing but the physical representation of an individual. So the genotype is partly contributing to the phenotype, but genotype is not the only reason why we are having different personality traits. There is also another component, which is the environmental factor. And this interaction of the genotype with the environment, that interaction is the one that is contributing to the phenotype. And this is what you call as a gene-environment interaction, which is contributing to all the phenotypic traits. The phenotype is not just your personality, your behavioral traits. It also includes all your disease-related outcomes like obesity or diabetes or cardiovascular disease, and also different health outcomes like your height, your weight, your BMI, your waist circumference. So all the phenotypic traits are driven partly by your genes and partly by your environment. So let me share some basics of genetics for those who do not have a background in genetics. So every eukaryotic cell has got a nucleus inside which you will find the genetic material, and the genetic material in humans is basically your DNA. So DNA, the human DNA is made up of 3 billion nucleotides, and the 4 important nucleotides are A, C, T, and G: adenine, cytosine, thymine, and guanine. So your entire human DNA is made up of 3 billion of these 4 nucleotides. And within these 3 billion nucleotides, you carry nearly about 40,000 genes, which have been discovered by the Human Genome Project. And what is the gene? Gene is nothing but a segment of DNA which carries the information for producing the proteins. So these proteins are produced from the genes. And what are these proteins? These proteins could be metabolic enzymes or enzymes carrying out various enzymatic reactions, catalytic reactions, and playing an important role in physiological pathways or metabolic pathways, or could be secretory proteins or membrane proteins or transport proteins. So they could be any kind of protein. So these are the proteins which are basically contributing to all the metabolic functions. So which means that if there is any defect or if there is any genetic variation, that can affect the quality and the quantity of the protein that is secreted. And if the quality and the quantity of the protein is affected, then that can have an impact on the metabolic functions. And if the metabolic activities are affected, then your physiological pathways, the metabolic pathways are also disrupted, and which will eventually disrupt the glucose homeostasis and eventually lead to the development of obesity and diabetes. So this is how the variations in the DNA can have an impact on the protein and in turn can affect the metabolic activities and lead to the development of obesity and diabetes. But as mentioned before, it is not just the genetic variations. There are also the environmental factors which will interact with the genes or the DNA or the genetic variation and contribute to the development of the disease. So what is basically a genetic variation? But before that, I'm going to ask you a question. There are about 3 billion chemical base pairs in human DNA. Approximately what % of these base pairs actually code for genes? So you can type your answers in the chat box. So there are 4 options for you: 100%, 30 to 50%, 1 to 2%, and 5 to 6%. So I'll give you 20 seconds. Just type the right answers in the chat box. So let's see who's getting the right answer first. What % of these base pairs actually code for genes? B. So I can see some answers B and some answers C. Good, there are different answers. So let me reveal the right answer. The right answer is basically C, which is 1 to 2%. So if you take the entire human DNA, it's basically just 1 to 2% of the DNA which carries the genes, which means that the genes which are producing the proteins. So the proteins are being produced only from that 2% of the DNA. The remaining 98% of the DNA is considered as a junk DNA, which has got the regions which are not coding for proteins. Okay, great. So now let's see what is a genetic variation. So say, for example, this individual has got this DNA sequence, and say this population has got 5 individuals. Okay, so you can see that 99.9% of the DNA is identical, where 0.1% carries all the genetic variations. And these genetic variations are the reason why we all looking different from each other. That's exactly what I was mentioning about the genetic heterogeneity. So you can see that some people carry T and some people carry A. So where there is a change from T to A. So T is a normal genotype and A is basically the genetic variation. So some people carry T and some people carry A, and there's a change from one nucleotide to another nucleotide. And this change from one nucleotide to another nucleotide is what you call as a SNP, or a single nucleotide polymorphism. You have to remember that our human DNA has got nearly about 3 to 4 million single nucleotide polymorphisms. And here I'm just talking about one genetic variation, one single nucleotide polymorphism. So there's a change from one nucleotide to another nucleotide. So depending on whether you carry T or A, you can classify individuals into 3 different categories as 0, 1, and 2. Okay, so if you do not carry any copies of the genetic variation, so if you look at this individual, he has got TT, which is the genotype. So there is no genetic variation. So that individual is classified as a 0 copy individual. If you carry 1 copy of the genetic variation and 1 copy without the genetic variation, then you're classified as a 1 copy individual. And if you carry 2 copies of the genetic variation, then you're classified as a 2 copy individual. So for a given genetic variation, you can either be a 0 copy or 1 copy or 2 copy. So there are about like 70 to 80 students over here. So if I try to genotype your DNA, and if I want to know like how many people are 0, 1, and 2, you will realize that some people are 0 copy, and some people are 1 copy, and some people are carrying 2 copies of the genetic variation. So in general, people carrying 2 copies, they have increased genetic risk. So here I'm talking about one genetic variation, one SNP. Remember, we all carry about 3 to 4 million single nucleotide polymorphisms. So which means that if you carry 2 copies in all the genetic variations, then what will happen? Which means that you're going to accumulate your genetic risk. Your genetic risk is going to increase. So what is a genetic risk? What do you mean by genetic risk? So let me explain that in a very simple way. So if you carry 0 copy, it means that low genetic risk. Remember, if you carry 0 copy, it doesn't mean that you have 0% risk of developing the disease. No, it doesn't mean that. 0 copy refers to low genetic risk, which means that you have decreased risk of developing the disease. So for a disease such as obesity or diabetes or cardiovascular disease, your risk of developing the disease is very low. If you carry 1 copy, it means that you have a moderate genetic risk, which means that you have moderate risk of developing the disease. And if you carry 2 copies, which means that you have very high genetic risk, and that means that you have increased risk of developing the disease. But this is exactly where the twist is. Just because you carry 2 copies, it doesn't mean that you are destined to become obese or diabetic. You can still overcome this genetic risk by modifying your lifestyle, which includes the healthy diet and doing more levels of physical activity. So by modifying your lifestyle and improving your healthy lifestyle, you can overcome the genetic risk of obesity or diabetes or cardiovascular disease. Could you please mute your microphones, please? Thank you. So even if you have a high genetic risk, you can still overcome the genetic risk by modifying your lifestyle. So this is the underlying basis on which we do the nutrigenetics and nutrigenomic research activities. So I have a question for you. Which of the following statements is incorrect about genes? A gene is a small piece of DNA. Genes are found in the nucleus. Genes are found on chromosomes. And genes are composed of DNA. So I have 4 options for you. Read the question carefully. Which of the following statements is incorrect about genes? So you can type your answers in the chat box. I'll give you 20 seconds. So we all typed B, genes are composed of DNA. Any other answer? Okay, let's go through the answers. A gene is a small piece of DNA. So gene is present in the DNA. So obviously gene is a small piece of DNA. So that's a correct answer. But my question is, which statement is incorrect? So A is not the right answer. So B, genes are found in the nucleus. So genes are obviously in the nucleus because genes are part of the DNA. So again, B is also not an incorrect statement. And genes are found on chromosomes. So chromosomes are nothing but a part of DNA, because DNA, when the cell undergoes cell division, at one particular stage of the cell division, the DNA becomes a chromosome. So obviously genes are part of DNA. So genes are also found on chromosomes. So that is also not an incorrect statement. Coming to the last statement, genes are composed of DNA. Obviously genes are present in the DNA. So genes are composed of DNA. So D is also not an incorrect statement. So all A, B, C, and D, all the statements are correct. So none of the statements are incorrect. So it's just a tricky question to confuse you guys. So I hope you understand that genes are part of the DNA and they are found in the nucleus, they are found on chromosomes, and they are composed of DNA, right? Okay, so moving on to the next topic, which is a complex disease. So this area of nutrigenetics and nutrigenomics is more applicable for complex diseases. So what is a complex disease? The diseases which are caused by many contributing factors. If you take obesity or diabetes or cardiovascular disease, they are not caused just by your diet. They're caused by lack of physical activity, lack of healthy diet, and also many genetic factors are involved. So that's why you call these diseases as multifactorial diseases. That is, multiple factors are involved in the development of the disease. And still, we do not know what are the genetic factors which are contributing. There are certain genetic factors which are contributing to the disease, but there are several other genetic factors which we still do not know, which are contributing to the development of the disease. So that's why we call these diseases as complex diseases. So let me give you a classic example of how type 2 diabetes develops. So if you take this individual, he has got a set of genes which are predisposing him to become insulin resistant. And he has also inherited some genes which are making him more hungry. So he ends up in consuming a lot of energy dense foods and not doing sufficient amounts of exercise. So during his lifetime, some of the genes which are responsible for producing the insulin, they become inactivated, and eventually that leads to the development of type 2 diabetes. So this is one pathway through which type 2 diabetes develops. But there are multiple other pathways which are contributing to the development of type 2 diabetes. So that's why we call these diseases as complex disease. It's not just one factor. There are multiple factors and multiple contributing factors which are leading to the development of the disease. So the area of nutrigenetics and nutrigenomics is more applicable for complex diseases, because the idea of nutrigenetics and nutrigenomics is basically to identify those factors which are contributing to the development of complex or multifactorial disorders. So I have another question for you. So which of the following diseases are multifactorial? Sickle cell anemia, coronary artery disease, cystic fibrosis, and colorectal cancer. So I'll give you 20 seconds. You can type your answers in the chat box. Which of the diseases are multifactorial? B, okay. I can see some answers B and one answer B and D. Any other answers? Okay, so let's go through the answers. So the first one is a sickle cell anemia. Sickle cell anemia is caused by one gene. So it cannot be called as a multifactorial because it's caused by a mutation in one gene. Just one factor is involved in the development of the disease. So you call sickle cell anemia as a monogenic disease. There's only one gene which is involved. Coming to the answer B, which is a coronary artery disease. Coronary artery disease is a cardiovascular disease, so which is classified as a complex or a multifactorial disease. Multiple factors are involved in the development of the disease. So coronary artery disease is a multifactorial disease. So B is the right answer. And C, cystic fibrosis. So cystic fibrosis is another monogenic disease, which is caused by a mutation in one gene. So again, cystic fibrosis is not multifactorial. And going to the last answer, D, colorectal cancer. So all types of cancer, they are multifactorial disease. So there's an involvement of genetic factor and the environmental factor together. So the right answer is B and D, coronary artery disease and colorectal cancer. So B and D is the right answer. So moving on to gene-environment interactions on obesity. So let's take obesity as an example, and let's have a look at the gene-environment interaction. So in terms of looking at the development of obesity, there are two important factors. So one is basically the lifestyle factors, which includes your diet, your physical activity, and various other lifestyle exposures like smoking, tobacco chewing, various factors which can contribute to the development of obesity. But at the same time, you also have your innate genetic factor, your DNA, which carries all the genetic variations and which can predispose individuals to become obese. But it's not just the genetic factor or the lifestyle factor, it's basically the interaction between the genetic and the lifestyle factors which contributes to the development of obesity. And that's why we say this: genes load the gun, but the environment pulls the trigger. This metaphor clearly explains the intricate interplay between the genes and the environment or the lifestyle factors. As I said, even if you carry the genetic risk, you can still overcome that genetic risk by modifying your lifestyle, that is modifying your environment, because the environment is the one that is pressing the trigger. It's not your genetic makeup. The genetic makeup, you carry that genetic risk right from your birth until the last year of your life. You cannot change your genetic risk, but what you can change is you can overcome the genetic risk by changing your lifestyle. So with this background, let's move on to what is nutrigenetics and what is nutrigenomics. So nutrigenetics refers to the study of the impact of the DNA sequence variations on chronic diseases such as obesity, diabetes, cardiovascular disease, or cancer in response to a particular diet. So let me explain this. For example, if you carry a high genetic risk, you're carrying 2 copies, you're having a very high genetic risk. And in addition to that, you're consuming completely unhealthy diet, consuming a lot of junk food, fried foods, and fast foods. So my question is, what is the risk of developing obesity for a person who has got a high genetic risk and consuming unhealthy diet? So in order to understand the risk of developing obesity, I need to look at the genetic makeup, and also I need to see the dietary intake. So the study of the interaction between your genetic risk and your dietary consumption, that study of the gene-diet interaction is what you call as a nutrigenetics area of research. Whereas nutrigenomics is completely a different area of research, because in nutrigenomics, as the name implicates, omics means expression, genomics means gene expression. So what is a gene expression? It's nothing but how the DNA is converted to RNA and how the RNA is converted to protein. So there's a change from DNA to RNA and RNA to protein. There's a flow of information. Information in the DNA is used for producing the protein. So that is what we call that as a gene expression, or the study of the gene expression is what you call as genomics. So omics is expression, genomics is gene expression, and nutrigenomics is nothing but how a particular nutrient is influencing your gene expression. The food that you're consuming has got all the nutrients, and these nutrients are being absorbed by the cells, and these nutrients enter into the cytoplasm, they go to the nucleus, they go and interact with your DNA and influences the conversion of DNA to RNA and RNA to protein. So your genes are expressing the protein in response to your diet. So that's why we say what you eat is what you are. So that is basically through this nutrigenomics area of research. So findings from nutrigenetics and nutrigenomics, they are essential for the implementation of the personalized nutrition. So personalized nutrition is where you're developing an optimum diet for an individual based on that individual's genetic makeup. So for example, if I carry a high genetic risk, what is the kind of diet I should consume? If I carry 1 copy, what is the kind of diet I should be consuming? So that is exactly what you call as a personalized diet. You're personalizing an individual's diet based on his or her genetic makeup. So as part of running these nutrigenetic studies, I'm leading 2 large collaborative networks. So one is called as a DCardia collaboration, which was initially funded by the British Heart Foundation. So the DCardia collaboration is basically focusing on the developed parts of the world, or in other words, focusing on the Western part of the world, Western population. So to date, we have nearly about 35 studies comprising more than about 100,000 individuals from the US, UK, and European countries. And in addition, we have access to the UK Biobank, where we have 500,000 people. And with access to various consortia-based studies, we have nearly about little less than 2 million people as a data set, like where we are running several nutrigenetic and gene diet interaction analysis. And these are some of the publications from the DCardia collaboration. But in addition to this collaboration, which is focusing on developed parts of the world, I'm also leading another large collaborative network, which is the GeNuIne collaboration. The GeNuIne stands for the Gene-Nutrient Interactions. And this collaborative network was initially funded by the British Nutrition Foundation. And the main objective of this collaboration is to implement nutrigenetic studies for the first time in lower middle income countries, that is focusing on the developing countries. So these are the countries in which I've implemented nutrigenetic studies for the first time. And what you see in the brackets, these are basically the funding organizations that have supported my nutrigenetic research activities in these lower middle income countries. Okay, fine. So nutrigenetics is nothing but the gene-diet interaction. So I'm going to share some interesting findings from the nutrigenetic activities in developed parts of the world and developing countries. So starting with the nutrigenetic studies, what is the status of nutrigenetic studies in developed countries in the UK, US, and European countries, that is in Western population? Okay, so it all started off with the discovery of the obesity gene. So this obesity gene was discovered in the year 2007 by two independent studies, two independent genetic studies. One was done in the UK population and the other one was done in the Spanish population. And both the studies discovered this obesity gene at the same time. And once this gene was discovered, people strongly believe that if they carry two copies of this obesity gene, they are destined to become obese. So people started to develop that fear in that mind. So in order to overcome this fear, genetic epidemiologists decided to test whether doing more levels of physical activity or consuming the healthy diet, can that overcome the genetic risk of obesity? So that was the point, like all the researchers started to explore gene diet interactions and gene physical activity interaction. So everyone were basically like very much interested in knowing whether doing more levels of physical activity or consuming healthy diet, can that overcome the genetic risk of obesity? Because people were having that kind of a fear, like if I carry two copies and I'm destined to become obese. So to clear this notion, this area of nutrigenetics and nutrigenomics started to gain a lot of attention, because nutrigenetics and nutrigenomics research work has been going on for the last 2 decades, but it gained a lot of attention, especially after the discovery of the obesity gene. So what is this obesity gene? The obesity gene is called, scientifically it's called the FTO gene. FTO is a genetic name for this particular gene, the gene name. So FTO stands for the fat mass and obesity associated gene. The genetic variations within this FTO gene have been shown to be strongly and consistently associated with obesity traits in various populations, in different ethnic groups, in men, women, adults, and also in children. So having seen such a strong effect of this FTO gene on obesity, the first question that comes to our mind is, can this effect be modified by a lifestyle factor such as physical activity or a dietary factor? So let me share some gene physical activity interactions and gene diet interaction. So I carried out a study in 20,000 people in the UK population, where I looked at the gene physical activity interaction on obesity. So these 20,000 people were stratified into 4 different levels of physical activity: active, moderately inactive, and inactive. And on the y-axis, you find the BMI. So my first question is, I want to know whether the genetic variant, this obesity gene variation, whether is it associated with BMI in all the levels of physical activity. So you can see that there are 3 colored bars: the white, yellow, and the green. So the white is 0 copy, and yellow is 1 copy, and the green is 2 copy individuals. And you can see that from this graph, you can see that individuals carrying 2 copies, they have significantly higher levels of BMI compared to 1 copy and 0 copy individuals. So if you carry 2 copies, you're having a higher BMI. And that's exactly what we identified in this study. And that was quite typical of all the levels of physical activity. But interestingly, the effect of the SNP on BMI was quite prominent among people who are leading an inactive lifestyle. So you can see that the effect is 0.44, whereas the effect for the other active group seems to be 0.2. So what is this 0.44? Which means that if you carry one copy, it means that you have 0.44 kilogram per meter square increase in BMI if you carry one copy. So which means that if you carry two copies, your effect is going to be doubled, 0.88 kilogram per meter square increase in BMI. So for individuals who are leading a physically inactive lifestyle, not doing any activity, no exercise, just sitting and watching television and not doing sufficient amounts of work, leading basically a sedentary lifestyle, so their effect is going to be 0.44 kilogram per meter square increase in BMI. Whereas people who are doing some level of activity, whether they are active or moderately inactive, you can see that the effect is 0.2. So which shows that doing some levels of activity or exercise can overcome the genetic risk of obesity, whereas a sedentary lifestyle can increase the genetic risk of obesity. So from these studies, we are able to clearly show, because following this study, this is one of the first studies which was published in 2009, immediately after the discovery of this obesity gene. But following these studies, there are several studies which have also confirmed this particular finding. Now, let me give you an example of how healthy diet can overcome the genetic risk of obesity. So we carried out a gene diet interaction study in European population. So we looked at 5 different European countries and we collected data of about 11,000 individuals. And these 11,000 individuals were followed up for nearly 5 to 6 years. And we looked at who are those individuals who are gaining weight and who are losing weight and who are still maintaining weight. So it was basically a longitudinal study design. And we carried out a meta-analysis of the data from these 11,000 individuals. And we identified a very interesting gene protein intake interaction. So you know that the European population, they do consume significant amount of meat products. So obviously the intake of animal protein is very high. And we know that consuming high amounts of animal protein can increase the risk of developing obesity and diabetes and even cardiovascular disease. So here in this graph, on the x-axis, you find the protein intake in percentage. On the y-axis, you find the weight change per year. And the 3 lines that you see, they're basically high genetic risk, moderate genetic risk, and low genetic risk, 0 copy, 1 copy, and 2 copy individuals. And you can see that as protein intake or protein consumption increases, the annual weight change also significantly increases for people who carry a high genetic risk. That is, if you are an European individual and you're carrying high genetic risk, that is 2 copies, and you're consuming high amounts of total protein, then that can increase the genetic risk of obesity. So in terms of translating this finding to personalized diet, so how can you provide a personalized diet? The best personalized diet will be lowering the protein consumption, that is less than 17% of the total energy intake. That will be an effective way to reduce the weight gain in those individuals who carry 2 copies. Of course, remember that proteins need to be consumed in significant amounts because you need proteins for performing all your activities, and protein consumption is definitely good, but reducing the animal protein consumption and improving or increasing the plant protein consumption is important. Another example from the same study, the same 11,000 people, but here we looked at the Mediterranean diet. The Mediterranean diet is considered to be a healthy diet, which is rich in omega-3 fatty acids, fruits, vegetables, and olive oil. So consuming a Mediterranean diet can help you to overcome the risk of becoming obese, that we all know. But here we did the same analysis at the backdrop of genetic susceptibility. So in this figure on the x-axis, you find the Mediterranean diet score, which means that higher the score, higher the consumption of the Mediterranean foods. On the y-axis, you find the weight change per year. The 3 lines that you see, they're basically low genetic risk, moderate genetic risk, and high genetic risk. And you can see that as the consumption of the Mediterranean diet score increases, the annual weight change decreases for people who carry a high genetic risk. That is, we know that if you carry a high genetic risk, you're going to gain more weight. But these high genetic risk people, by consuming more amounts of Mediterranean foods, they were able to reduce their weight over a period of time. So which clearly shows that even if you carry a high genetic risk, by consuming a healthy diet such as a Mediterranean diet, you can overcome the genetic risk of weight gain. So all these studies, these are just few studies that I've given you as examples, but there are plenty of studies in developed parts of the world. So the status of nutrigenetics is very well established in the UK, US, and European countries. So there's no question of like what is lacking or what is missing. Of course, there are several challenges as part of doing nutrigenetic studies in developed parts of the world, but let's move on to developing countries where there are a lot of limitations and challenges in conducting these nutrigenetic studies. So what is the status of nutrigenetics in developing countries or in lower middle income countries? And that is exactly the reason why I started this GeNuIne collaboration. So GeNuIne stands for the gene-nutrient interaction. The main objective is basically to implement nutrigenetic studies in contrasting ethnic groups. The first nutrigenetic study was implemented in the Indian population, then in Sri Lanka and Indonesia and Brazilian population, Ghanaian, that is the West African population, Turkish population, and now in Peruvian population. And also we are running some studies in other lower middle income countries, in addition to the ongoing collaborations in developed parts of the world. And these are some of the publications from the GeNuIne collaboration. So let me share some interesting findings from each of these lower middle income countries. The first nutrigenetic study was implemented in the Asian Indian population in the year 2017. And where we found a very significant interaction between your genetic risk and carbohydrate intake. So you know that for Asian Indians or any South Asian population, the main source of energy comes from the carbohydrate, whereas in the Western parts of the world, it's basically the fat consumption. So here in South Asian population, given that the rice is the main source of energy, and we found a very interesting interaction with the carbohydrate intake. So here you can see on the x-axis, you find the tertiles of carbohydrate intake. On the y-axis, you find the odds ratio for obesity, which is nothing but what is the risk of becoming obese. And the red bars are those individuals who carry a high genetic risk. And you can see that people who carry a high genetic risk and consuming more than 500 grams of carbohydrate per day, they had nearly about 2.46 times increased risk of obesity. So which means that if you are an Asian Indian and you're carrying a high genetic risk and you're consuming high amounts of carbohydrate, then your risk of obesity, your genetic risk of obesity will increase by 2.46 times. So which is a very clear indication of the fact that the Asian Indians, one way to prevent obesity in Asian Indian population would be to reduce the consumption of carbohydrate, that is less than 300 grams of carbohydrate per day. Let me give you another example of another South Asian population, which is the Asian Sri Lankan population. So Asian Sri Lankans, again, they do consume significant amount of carbohydrate, but the way they cook and the way they eat is so different from that of the Asian Indian population. So let me share the first nutrigenetic study that we implemented in the Asian Sri Lankan population. So again, we found an interaction with the carbohydrate intake, but here on waist-hip ratio. So waist-hip ratio is basically an indicator of central obesity. So there are two types of obesity. One is a common obesity and the other one is a central obesity. So the common obesity indicator is basically the BMI, whereas for central obesity, the indicator is either waist circumference or the waist-hip ratio. So central obesity is a very important risk factor for the development of cardiovascular disease. And here, all these studies that I've done in Asian population, majority of the studies have shown an interaction on central obesity and its indicators. And here we found an interaction on waist-hip ratio. So here on the x-axis, you find the tertiles of carbohydrate intake, low, medium, and high consumption of the carbohydrate. And the red bar, people with high genetic risk, and the white bar, individuals with low genetic risk. And you can see that those Sri Lankans who are consuming high amount of carbohydrate, again, more than 500 grams of carbohydrate per day, and carrying a high genetic risk, they had significantly higher levels of waist-hip ratio compared to individuals with low genetic risk. So, which clearly highlights the fact that again, Asian Sri Lankan population, in order to overcome the genetic risk of obesity, their consumption of carbohydrate should be lesser than 300 grams of carbohydrate per day. So we also carried out a study in the Indonesian population, which is the first nutrigenetic study of its kind in a Southeast Asian population. This study was done under the funding from the Newton Fund, and we focused on the Minangkabau community living in the West Sumatra island. And Minangkabau is the world's largest matrilineal community. And we looked at the Minangkabau women as part of this nutrigenetic study. So we found a very significant interaction with the protein consumption. And you know that Indonesians do consume significant amounts of meat products, and consumption of the animal protein is very high. So in this graph on the x-axis, you find the tertiles of protein consumption. On the y-axis, you find the waist circumference, and the tertiles are low, medium, and high consumption of protein. So the low is basically where they were consuming about 70 to 75 grams of protein per day, and high is where they were consuming about 140 grams of protein per day, which means that double the amount of what is required for the Indonesian population. But interestingly, we found an interaction involving people who are consuming low amounts of protein per day, that is around 70 to 75 grams of protein per day. So here we found that those individuals who are consuming low amounts of protein and carrying a high genetic risk, they had lower waist circumference, which is another indicator of central obesity. So which clearly highlights that even if you carry a high genetic risk, by consuming low amounts of protein, you can overcome the genetic risk of central obesity. But you have to understand the public health message very carefully. I'm not asking you to reduce your protein consumption. My message is that you have to reduce the consumption of the meat or the animal protein consumption and improve your plant protein consumption. So that is a way by which you can overcome the genetic risk of obesity or the central obesity for this particular Indonesian population. But interestingly, we also carried out a study in the West Asian population, so which is the Turkish population. So there we found another interesting interaction again with the total protein intake. So even the Turkish people, they do consume significant amount of meat products, and animal protein consumption is very high in this population as well. But of course, the way they cook is so different from that of the Indonesian population. So, but of course, we did not look at the cooking patterns comparing the two populations. We just looked at the dietary intake, but that will be a very interesting component to explore, like how the cooking styles and cooking patterns can have an impact on these findings. So here we found that there was an interaction with the protein consumption, again with central obesity. That was very interesting. So we found that those individuals who have a high genetic risk and those who have a high genetic risk and consuming high amounts of protein-rich foods, they had 3.3 times increased risk of central obesity, implicating that consuming an unhealthy diet such as high amounts of animal protein and carrying a high genetic risk can increase the risk of becoming centrally obese. So all these findings are highlighting the fact that a healthy lifestyle can overcome the genetic risk of obesity or central obesity. And another interesting finding, like which we published last year, which is about adiponectin. So adiponectin is an important biomarker of obesity. So having good levels of adiponectin can prevent you from becoming obese. And we found that this circulating adiponectin in your body could be a link between your genetic risk and cardiometabolic health. So we carried out a study in Asian Indian population where we looked at 2,000 individuals. And we found that those who had high genetic risk, they had increased risk of cardiometabolic diseases. And interestingly, people who had high genetic risk, they had increased risk of cardiometabolic diseases even after adjusting for all the confounding factors. And these people who had high genetic risk, they also had significantly lower levels of adiponectin concentration. So if you look at this graph, you can see that people carrying 1 copy or 2 copy individuals, those who have high genetic risk, they had significantly lower levels of adiponectin concentration. And individuals who had low levels of adiponectin concentrations, they had increased risk of cardiometabolic diseases. Now, the main question is whether I wanted to know whether the circulating adiponectin concentration is the reason why we are seeing an association of genetic risk with cardiometabolic diseases. Or in other words, I wanted to know whether adiponectin is a mediator between your genetic risk and cardiometabolic diseases. So what I did, I tested for the association of the genetic risk and cardiometabolic diseases again, but now adjusting for the adiponectin. But after adjusting for adiponectin, I found that there was no association. So which means that the association of the genetic risk with cardiometabolic diseases is being mediated through adiponectin concentration. So it's only because of the low levels of adiponectin, your genetic risk is increasing. So how is this finding is going to help me in providing a personalized diet? So if I'm going to screen your DNA, if I'm going to see your genotype, and if I know that you have a high genetic risk, then the best way to prevent the development of cardiometabolic disease would be to improve your adiponectin levels in your body. How can you boost your adiponectin levels? So one way to boost your adiponectin levels would be basically to consume more amounts of omega-3 fatty acids, so which is kind of more of a healthy diet, and also basically to improve your physical activity levels as well. So I'm not able to move this. Yeah, so by improving your physical activity levels and consuming a healthy diet such as omega-3 fatty acids, so that would be a best way to improve your adiponectin concentrations as well. So in fact, there are several studies which have shown that there are some medications which can help you to boost your adiponectin concentration, but I would start with the lifestyle modifications to begin with to improve your adiponectin levels, which in a way can overcome the genetic risk of cardiometabolic diseases. So in terms of looking at the summary, so the GeNuIne collaboration provides the first evidence of all these gene diet interactions in these populations. So if you need to implement a personalized diet for these people, you need to take into consideration of the findings from each and every ethnic group, like South Asia or West Asia, Southeast Asia. You cannot use a finding from South Asian population to implement that for the West Asian population. You have to use the findings from South Asia to implement personalized diet only for South Asians, because we all carry different genetic makeup. So we cannot use the findings from one population to another population. That's the reason why I'm doing a nutrigenetic study in each and every ethnic group, so that the findings from that population can be used for implementing a personalized dietary strategy to prevent obesity in those populations. So I have a question for you now. So the role of the DNA sequence variation in response to nutrients is precision nutrition, nutrigenomics, personalized nutrition, and nutrigenetics. So which is the right answer? You can type the right answer in the chat box. The role of the DNA sequence variation in response to nutrients. I'll give you 20 seconds. I can see the answer D. Are there any other answers? There's also one answer B. B. Okay, most of the answers are B. So here the question is like the study of the role of the DNA sequence variation. I'm talking about your genetic variation. So genetic variation is basically your 0 copy, 1 copy, and 2 copies. So in response to a nutrient, how is your DNA reacting to it? How, whether you have high genetic risk or low genetic risk. So that study of gene diet interaction is basically nutrigenetics. So the right answer is D. So D refers, which is the nutrigenetics is the right answer, which is the study of the role of DNA sequence variation in response to nutrients. So moving on to the pathophysiology of type 2 diabetes. So when you look at type 2 diabetes, which is another important complex trait, you can see that there are 3 important factors. One is genetics, epigenetics, and environment. So let's just focus on genetics and environment now. So the interaction of these 3 factors is the one that is contributing to the development of type 2 diabetes. Now let's focus on the gene and the nutrient, which is what we call as a nutrigenetics, gene-nutrient interaction. So there are studies which have shown the impact of the nutrient intake on type 2 diabetes. And there are also studies which have shown the impact of the genetic variants on type 2 diabetes. But the key question is, what is happening with the interaction between the genetic factor? So what is happening with the interaction between the genetic factor and the dietary factor on the risk of developing type 2 diabetes? So that's what I'm going to show you now. So let me share a very interesting study on the gene diet interaction in relation to the first nutrigenetic study that was done in the Indian population in relation to type 2 diabetes. So this TCF7L2 is a very important candidate gene for type 2 diabetes. And it is a gene that has been discovered for type 2 diabetes in Western parts of the world. So we wanted to see whether there is any interaction on type 2 diabetes. But interestingly, what I identified was an interaction with the dietary fat consumption on high density lipoprotein cholesterol. That is, those individuals who are having a high genetic risk and consuming high levels of unhealthy diet, they had low levels of high density lipoprotein cholesterol. So high density lipoprotein cholesterol is considered to be a healthy cholesterol, so which is basically you need to have high levels of that good cholesterol. So the ideas behind, like, how, if you have a high genetic risk, how can you improve your good cholesterol levels in your body? So let me share that interesting finding. So let's have a look at the interaction between this TCF7L2 single nucleotide polymorphism and the fat consumption on HDL cholesterol. So first, we looked at the link between the dietary fat and the HDL cholesterol. There's no genetics over here. We just looked at the dietary fat consumption and HDL cholesterol, and we found that the p-value association was not significant. So there's no link between the dietary fat consumption and the high density lipoprotein cholesterol. But when you include the genetic component in this model, so here you can see that on the x-axis, again, you have the tertiles of dietary fat, low, medium, and high. On the y-axis, you find high density lipoprotein cholesterol. The red bar, people with high genetic risk, and the blue bar, low genetic risk. And you can see that those individuals who are consuming low amounts of dietary fat, despite carrying high genetic risk, they had high levels of good cholesterol. So which means that even if you have a high genetic risk, by reducing your fat intake, that is reducing the intake of the fried foods or oily foods and consuming a healthy diet, that can improve your good cholesterol level and overcome the genetic risk. So we did not only see that with the total fat consumption, we also found a similar interaction with the PUFA, polyunsaturated fatty acid intake. So here you can see that there was no link between the PUFA consumption and high density lipoprotein cholesterol. But when we included the genetic factor into the model, we found that those individuals who are reducing the PUFA consumption, and despite carrying a high genetic risk, they had high levels of good cholesterol. So which clearly shows that reducing the intake of PUFA or reducing the total fat consumption, that can improve your good cholesterol level by overcoming the genetic risk. So what is that gene diet interaction over here? So we know that TCF7L2 is a gene for type 2 diabetes and not for HDL cholesterol. So there was no association. And likewise, HDL also did not show any association with fat intake or the PUFA intake. But the interaction is, if you have a high genetic risk and you're consuming reduced levels of fat consumption, then which means that you can have high levels of good cholesterol. You can improve your good cholesterol levels. But if you have a high genetic risk and you're consuming high amounts of fat-rich foods, then it means that you're going to have low levels of good cholesterol. So this is exactly what you call as a gene diet interaction. That is how your lifestyle or a dietary factor can overcome the genetic risk of obesity or diabetes or even cardiovascular disease. The cholesterol level is an indicator for cardiovascular disease. So this is what you call as an interplay between the nutrition and the genome. So if you're interested in knowing more about like what I'm doing in other parts of the world as part of the GeNuIne collaboration, feel free to go into this publication. This was published in the year 2020. But quite recently, I've also published another paper focusing on vitamin B12 and vitamin D deficiencies. I'm not going to go into those findings, but if you're interested in knowing, that paper is available online. So feel free to go into that, and that will give you an idea about like what are the different nutrigenetic studies that I've done in relation to B12 and vitamin D deficiencies. So now let's move on to nutrigenomics. So we have seen a lot about nutrigenetics. Now let's have a look at nutrigenomics. So as I said, nutrigenomics is nothing but the study of the role of the nutrients in gene expression. So it all starts with your diet, your food that you're consuming. So the food that you're consuming has got all the nutrients, the macronutrients, carbs, protein, fats, fiber, and also all the micronutrients like vitamin D, vitamin B12, flavonoids, polyphenol. So all these nutrients eventually need to be absorbed by your cells. So these nutrients are being absorbed by the cell, and these nutrients, they enter into the cell where they reach the cytoplasm. But once in the cytoplasm, there is a molecule which is eagerly waiting for the nutrient to enter the cytoplasm. And that molecule is nothing but the transcription factor. The transcription factor plays a very, very important role. I'm just seeing a message. I just have 5 minutes left for my presentation. Oh, okay, I have a lot to cover. Anyway, I'll see what I can do. Is it okay if I go on for another 10 minutes?
Okay, that's fine.
Yeah, okay, fine, great. So the transcription factors, they play a very important role. So one role is basically where it goes and binds to the nutrient. And the other role is basically to take the nutrient to the DNA, where it binds to the DNA, interacts with the DNA, and convert the DNA to RNA and RNA to proteins and proteins to metabolites. So in response to a particular nutrient, there are certain RNA molecules which are produced, and that study of the expression of the RNA is what you call as a transcriptomics, that is RNA transcript expression. Likewise, there are certain proteins which are produced in response to nutrient, and that study is called as the proteomics, expression of the proteins in response to nutrients. And likewise, metabolomics, study of the expression of the metabolites in response to nutrients. So all these areas of omics, transcriptomics, proteomics, metabolomics, they all come under one big umbrella, which is the nutrigenomic area of research. And that understanding the differences in the gene expression is required for preventing the disease, because these differences in the gene expression, they serve as molecular biomarkers. And identifying these biomarkers is required for preventing the development of the disease. How can you prevent the development of the disease? Will you please mute your microphones, please? Yeah, thank you. So how can you prevent the development of the disease? So one way to prevent the development of the disease is developing and implementing the strategies for personalized nutrition. So the personalized meal plan is developed in a way taking into account of your gene expression level. So one is basically to look at your genetic risk, looking at whether you carry 2 copies or 1 copy or 0 copy. On the other hand, looking at your gene expression level, whether your genes are upregulated or downregulated, and based on that, providing a personalized meal plan. So I have another question for you. The study of the role of nutrients in gene expression is precision nutrition, nutrigenomics, personalized nutrition, and nutrigenetics. So you can type your answers in the chat box. Is it A, B, C, or D? I can see all the answers as B. Definitely, yes, the nutrigenomics is the right answer, which is the impact of the nutrients on gene expression. That's right. So B is the right answer. So we have seen the genomics, transcriptomics, proteomics. Let's have a look at metabolomics. So metabolomics is a very important area of research where we are looking at the metabolites, which are the end products of the entire metabolism. So looking at the metabolites will give you a clear reflection of what your dietary pattern is like. So metabolic profiling is very important to understand the molecular basis of health and disease. So how can you do the metabolic profiling? So for example, depending on your question, you could do a human study or an animal study, or you can collect the blood sample and get the serum or plasma, or you can also get the urine samples or fecal samples or even breast milk. So again, it comes back to the question that you have as part of the metabolic profiling. And what methodology can be used? So again, depending on your question, you can use mass spec or NMR or different kind of a chromatographic technique. So for example, if I'm looking at 1,000 urine samples, so from each urine sample, I'm going to get 3,000 metabolites. So for 1,000 urine samples, I'm going to see like a huge amount of data. So how am I going to handle this data? So for that, you definitely need to have a sophisticated software, and data analysis is very crucial. So for example, you see different peaks over here. So each peak is representing a metabolite, whether you're going to have high levels of metabolite or low levels of metabolite. So by knowing whether you have good levels of the healthy metabolite or whether you're having very high amounts of bad metabolite, so accordingly, it gives you an opportunity to change your dietary pattern. So this is how you translate the findings from metabolomics to precision nutrition or the personalized nutrition. So we have seen nutrigenetics, nutrigenomics, and metabolomics. So let's have a look at briefly about the epigenetic modifications as well. So epigenetics is basically a non-genetic area of research where we are looking at the changes in the gene expression. So where there's a change in the phenotype, but there's no change in the genotype. So one of the most important epigenetic modification is your DNA methylation, where one of the nucleotides, which is a cytosine, gets methylated. So how is this methylation taking place? It's taking place in response to your diet, the food that you're consuming. It should have good amounts of methyl donors. So which means that healthy diets are quite rich in methyl donors, which are responsible for methylating the cytosine. So how is that regulating the gene expression? For example, if you're not consuming a healthy diet, then your DNA is not methylated. So if the DNA is not methylated, the transcription factors can bind and initiate the gene expression, and that can lead to overexpression of the genes, which is not healthy for you. Whereas if you're consuming a methyl-rich diet, a healthy diet, then the methyl groups are added on to the cytosine. That will prevent the transcription factors binding to the DNA, and that results in gene silencing. That will stop the gene expression. So we all carry several genes, but we don't need all these 40,000 genes to be active or continuously expressing the protein 24/7. We just need them only when they are required. For example, when you're consuming any food, you need these genes to activate. So only then you need that. Other times they could be silenced. But if that is not happening, then that can lead to obesity, diabetes, and cardiovascular disease. And there's a very strong epigenetic link between maternal diet and fetal growth. So studies have clearly shown that unbalanced maternal diet during pregnancy can have an impact on fetal growth, and that can result in the development of cardiovascular disease when these infants become adults. And likewise, seasonal variations in maternal methyl donor nutrient intake during the periconceptional period can have an impact on the maternal biomarkers, and which can lead to differences in the methylation patterns. And likewise, obesity during pregnancy or diabetes during pregnancy can also have an impact on the DNA methylation changes. And lastly, the micronutrient deficiencies can also have an impact on the methylation changes. So we carried out a vitamin D pregnant mother cohort study in Indonesia a few years ago. So where we found that pregnant mothers in the third trimester and those who have a high genetic risk, they had significantly low levels of vitamin D. So these pregnant mothers who had vitamin D deficiency during the third trimester, they gave birth to babies which had a small head circumference that is less than 35 centimeter. So we know that the babies with small head circumference at birth, they are at a higher risk of developing cardiovascular or cardiometabolic diseases when these infants become adults. Given that strong correlation, so we can clearly see that the vitamin D supplementation during pregnancy can prevent the abnormalities in the newborn babies. So, which is clearly coming back to the fact that a healthy lifestyle can overcome the genetic risk of obesity or various other chronic diseases. So foods rich in methyl donors are very important. That's why we say what you do and what you eat in the first 1,000 days of your life, it plays a very crucial role for the rest of your life. So I have another question for you. Which of the following diet-derived compounds have epigenetic effects? One is the butyrate, curcumin, genistein, and none of the above. So I think I'm running out of time, so I'll just reveal the answer. So in this butyrate, curcumin, and genistein, All the three have epigenetic effects. So butyrate, curcumin, and genistein, they can all have induce epigenetic modification, which is the DNA methylation. They can all methylate the DNA, so they are all required as part of your diet, and they're quite rich in methyl donors. So going to the last part of my talk, which is about the impact of the diet on gut microbiome. So gut microorganisms plays a very important role because the food that you're consuming, it's not only interacting with your DNA, not only interacting with your gene expression or contributing to the metabolite, but also they play a very important role in reaching your gut lumen, where you carry 2 kilograms of the gut microorganisms. So the gut microorganisms play a very, very important role. So let's have a look at the role of the gut microbiome in health and disease. So if you're consuming an unhealthy diet of foods which are rich in sugar or saturated fatty acids and consuming high amounts of protein, they can all reduce the production of the short chain fatty acids. The short chain fatty acids are very important for maintaining your cognition level, your mental health, your physiological activities, and most importantly, in boosting your immune system. So if your short chain fatty acid levels are going down, then that can result in making you a victim of several infections. That's the reason why during the COVID pandemic, a lot of people were asked to improve the production of short chain fatty acid, that is to nourish your gut microbiome. And also there is increased production of the TMAO. TMAO is basically, this trimethylamine, so which is basically a chemical which is present in when you consume too much of meat products or dairy products. The trimethylamine in these products get oxidized to produce a trimethylamine oxide. So high levels of this particular molecule can lead to the development of cardiovascular disease as well, and inflammation in the gut, leading to inflammatory bowel disease and also gut-related health problems. But on the other hand, if you're consuming a healthy diet, like the diet which is rich in fiber or probiotics, that can improve your short chain fatty acid production and also improve your lipid metabolism and prevent you from developing diabetes by improving your insulin sensitivity, and most importantly, boosting your immune system and reducing the risk of all the infections. So gut microbiome is also very equally important, because studies have clearly highlighted the fact that 90% of all the type 2 diabetic cases worldwide is purely because of the gut microbiome dysbiosis, because the gut microbiome composition. The gut microbiome is not only composed of bacteria, but also you carry viruses, phages, and yeast. And gut microbiome composition plays a very important role in terms of preventing you from developing such chronic diseases. So what percent of all the cells in the human body are bacterial cells? Please remember that your bacterial cell content is nearly more than 70% in your body. So gut microbiome plays a very, very important role in contributing to the health outcomes. So personalized nutrition. So what is personalized nutrition? It's basically taking into account of your diet and your physical activity. And based on that, how your genetic risk could be estimated. And based on that, you're developing a personalized diet for an individual. So if you carry 2 copy or 1 copy, what is the kind of diet you should be consuming? So this is what has been happening 10 years ago in terms of developing a personalized diet. But now we stopped using the term personalized nutrition. We have started using the term precision nutrition, purely because of the fact that the food that you're consuming, it's not only interacting with your DNA, which is what we have seen as nutrigenetics, but it is also interacting with the metabolites, which is what we are seeing as nutrimetabolomics area of research. And also your food is going into the gut lumen, where we are seeing the gut microbiome interaction. And last but not least, which is the epigenetic modification, how the food is influencing the DNA methylation and various other epigenetic modifications. So as soon as you consume any food, the food is going into 4 different compartments and they are acting in parallel. So if you need to implement a personalized diet for a person, you need to look at it as a 4-way interaction, as metagenome, hyperbolome, epigenome diet interaction. And understanding this interaction is crucial for the implementation of the precision nutrition. That's why we use the term precision nutrition, because we are taking into account of this holistic approach of looking at nutrigenetics, metabolomics, gut microbiome, epigenetics, but in addition to various other phenotypic traits, like looking at your meal content and meal timing, and also looking at your sleep patterns, your socioeconomic status, your age, your sleep patterns, and various other factors taking into account. So we are trying to implement a personalized diet taking into account of various factors put together. And this is what I call as a revolution in the field of nutrition, how a personalized diet for an individual could be designed in a very precise way, taking into account of various multiple factors. So this is how I see a transition or a translation from nutrigenetics to precision nutrition. So on this note, I would like to thank all my collaborators from the GeNuIne collaboration, also from the dCardia collaborators. And finally, thank you all so much for listening to my talk. And if you have any questions in future, you can drop me an email at v.karani@reading.ac.uk. And I'm available on Facebook, LinkedIn, and Instagram and Twitter as Vimal Karani. And now I'm happy to take any questions. Thank you.
Thank you, Professor Vimal, for the great talk and very insightful for us. So we already have some questions in the chat box, but before we are going to those questions, I'm curious to know about your study in Indonesia. Why are you interested in seeing the interaction between the protein and obesity incidence or prevalence in Indonesia instead of carbohydrate, let's say, because we are also consuming a lot of carbohydrates here. Rice is our staple food. Maybe you can explain a little bit to us.
Yes, so we didn't specifically look at protein intake. So for any country that we looked at, these were the interactions which turned out to be significant. For example, when we did the study in Indian population, we found an interaction with carbohydrate intake, but we did look at the interactions with protein consumption, fat consumption, fiber consumption, but none of those interactions were significant. It was only the carbohydrate interaction. So likewise, for the Indonesian population, we did look at the interaction with carbohydrate, protein, and fat, but this protein intake interaction turned out to be significant. Hello, are you there? I think she has got some internet connectivity issue. Okay, while she is coming back, let me try to answer some of the other questions in chat. So I would like to ask a question. You mentioned the gene diet interaction obesity traits have been, wait a minute. You mentioned that gene diet interaction obesity traits have been studied in Asian population and separately, and so as to create. I was wondering if there's a possibility in developing algorithm to predict these, regardless of ethnicity and race. Would such an inter-ethnic analysis would be useful to predict gene diet interactions more accurately? That's a very interesting question. So basically, you have to understand that ethnicity plays a very important role, and we are in a stage where we still do not have the clear picture of the genetics. So for example, if you take obesity, only 4 to 6% of the genetic variations have been identified. So which means that the remaining 94, 95% of the genetic variation for obesity still haven't been explored and we still haven't discovered yet. So which is very, very important. So we need to make sure that we identify all the genetic variations before we move on to the level of personalized nutrition, because personalized nutrition is very much in an infant stage. And we need to work on it in terms of how that could be translated to precision nutrition. But having said that, with the data that is available at the moment, there are a lot of researchers from the field of bioinformatics and big data analysis. They are trying to do kind of artificial intelligence or the machine learning approaches, and how they are using those machine learning approaches, like you have supervised learning and unsupervised learning, and developing algorithms and models, and how you can, for example, we did a study at the University of Reading, which is called the Food for Me study, and where we looked at the European population and we kind of developed this kind of an algorithm. So we just looked at one genetic variant. For one genetic variant, they were about like nearly about more than 300 different models of personalized diets that we were able to develop. So for one genetic variant, you can have more than 300 different algorithms that could be developed. So just imagine we all carry about 3 to 4 million genetic variations. So the number of algorithms and models that are going to be developed required a sophisticated software, and that is a level of precision that we are looking for. And a lot of research is going on in this field. And as I said, the genetic variants need to be identified. And these genetic variations are not going to be the same for all ethnicities. For each ethnic group, there are going to be a set of genetic variation, because one genetic variation might be showing a susceptible effect. For example, we looked at a PPAR gamma gene, where we found that for Indian population, this gene was not showing any impact at all. Whereas in Caucasian population, in the US population, we found that the gene was showing a very strong susceptible effect for type 2 diabetes. So there are some ethnic specific genetic variations. And of course, like we are doing kind of like inter-ethnic meta-analysis and trying to do some, but the level of heterogeneity, the genetic heterogeneity that I mentioned in the beginning of my lecture, that was very high. So it would be very difficult to develop one personalized nutrition or diet for the entire world, because we are not talking about a universal dietary recommendation here. We are all moving away from one size fits all approach towards a personalized approach.
All right, thank you, Professor Vimal, for answering, I think, Payal's questions. I'm sorry, I'd had some technical difficulty. Yeah.
I hope you got the answer from my question, for your question. Yeah, okay, good.
Thank you. And let's continue. There are other questions. Let's see question from Dr. Siti Muslimatum. So he asked, in Sri Lanka study, why those with low risk genetic and low intake of carbohydrate have high prevalence of high waist hip ratio?
Sorry, which question? Yeah, in Sri Lanka study, why those with low genetic risk and low intake of carbohydrate have high prevalence of high waist hip ratio? Mm-hmm. Well, see, when you look at the graph, like the main concern is for people who have a high genetic risk. So whenever you work in the field of nutrigenetics, my concern as a public health epidemiologist will be looking at people who have high genetic risk, because I'm trying to give a public health message for people who have a high genetic risk, not for people who have a low genetic risk. Because a person who has a low genetic risk, the first question that comes to his mind would be, I have low genetic risk, so can I consume whatever I want? So you can't give that public health message saying that you can consume whatever you want, because here I'm talking about 1 or 2 genetic variations. So that individual might be a low genetic risk for those genetic variations. But on the other hand, as I said, we all carry multiple genetic variations. So he could be a genetic risk for other genetic variations, which we haven't screened. So we cannot provide a kind of a message or a take-home message for low genetic risk people. So even if you read that particular paper which I published, I would have clearly mentioned that my concern is only for people who carry a high genetic risk as part of my study. So I want to give the public health message, reduce your carbohydrate intake so that you can overcome that high genetic risk. So obviously you cannot judge a person who has got a low genetic risk, because he might be high genetic risk for other genetic variations.
Okay, thank you, Professor Vimal. Hopefully that answers the question from Dr. Siti. She has another questions, but I will continue to a question from other participant, from Ms. Nurul Dina Rahmawati. She mentioned, thank you for a very enlightening presentation. And her question is, in Gene Mediterranean diet study, would you please explain more on how long was needed to observe the weight reduction, and whether the weight loss maintenance was also observed, and why we have not got much discussion on the fat intake? Thank you.
Okay, thank you for the question. So basically that study was done as part of this pan-European program, which is called as the Diogenes Study. Diogenes stands for the diet, obesity, and gene. And the main objective of that consortium was basically to look at the interaction between the genetic and the dietary factors in relation to weight gain. So this study was done as part of the EPIC consortium as well, EPIC cohort, and where these 5 European countries, and we followed up for a long time. And so the Mediterranean dietary analysis was only a part of that entire study. So we looked at various dietary factors, and there are various publications which were published from the GeNuIne study. And in fact, like we also found an interaction with fat consumption. In fact, like if you look at the FTO gene, majority of the FTO gene diet interactions were pertaining to the fat consumption, because as I said, for South Asians, the main source of energy comes from the carbohydrate. Whereas for the European population and the Western part of the world, the main source of energy comes from the fat consumption. So we did find a significant interaction with fat consumption, where people were consuming high amounts of fat-rich foods and carrying the two copies, they had increased genetic risk of obesity. We did see that, and in terms of longitudinal study as well as a cross-sectional study. But yeah, in this study, of course, like, I mean, because of the time restriction, I didn't show those findings. I just restricted to the protein consumption and the Mediterranean diet. And also it depends on the question, how long you would need to see the impact on the weight loss or anything relating to the anthropometric measurement. So it depends entirely on your question. But this study was basically wanted to look at the weight gain. But here, as part of the GeNuIne study, we did not look at just the gene diet interaction, because that cohort, that data set was used for various analysis. There are like more than 30 publications which came out from the GeNuIne study. So just drop me an email. I can send you the link to all those studies and how that longitudinal study was planned.
Okay, thank you, Professor Vimal. I will just continue to the next question from Ms. Arimatea Arwani. She would like to know if there is someone who has high risk of obesity and has a bad lifestyle, eventually the person got the obesity. But if their lifestyle changed, is it possible to cure the obesity?
See, obesity, diabetes, cardiovascular disease, they are all chronic diseases, which means that once you develop the chronic disease, you have to live with that chronic disease. But what you can change is, for example, if you have diabetes, you can maintain the sugar levels, blood sugar levels. And if you're cardiovascular disease, if you have high blood pressure, then you can be on a medication to maintain your blood pressure levels. And the same applies for obesity as well. So you have to maintain your lifestyle to make sure that you're overcoming that genetic risk, because you're developing obesity, diabetes, cardiovascular disease, partly driven by your diet and partly driven by your genetic risk. You can't do anything with your genetic risk, because genetic risk from day one until the last day of your life, you have to live with that genetic risk. But what you can change is your lifestyle. But once you maintain a healthy lifestyle, you can overcome that genetic risk and sustain that weight as long as you're maintaining that healthy lifestyle. Because the moment you shift to the unhealthy lifestyle, your genetic risk will start to play again. So which means that you can become obese again.
Okay, thank you, Professor Vimal.
I think there's a question about...
Yeah, there is another question, last question maybe. In relation, this is also from Dr. Siti Muslimatun, in relation to the finding about TCF7L2 gene and the fat intake with the HDL level, what is your suggestion whether or not we should have gene testing to understand our risk?
See, as I said, I have to state that this study was done in the Asian Indian population. So this is definitely not applicable for Indonesian population. And of course, we have done some studies in relation to TCF7L2 gene, and not just on TCF7L2, we have also done some studies on FTO, TCF7L2, and various other genetic variation in Indonesian population. So have a look at those publications from GeNuIne collaboration. But with regards to this question, I would definitely not take it for testing at this point, because HDL level is a very complex trait and it's driven by various other genes. TCF7L2 is a gene for type 2 diabetes, but it was an accidental finding that we found an interaction on HDL cholesterol, because we were expecting HDL cholesterol to be an important outcome when we are looking at lipid-related genes like CETP or lipoprotein lipase or ApoE, which are very important genes for total cholesterol or HDL cholesterol, all the lipid traits. But here, like, we found an interaction of the TCF7L2 with fat consumption on HDL cholesterol, which is an accidental finding, and this needs to be definitely replicated before it can go to the level of genetic testing.
Okay, thank you, Professor Vimal. I think we have another 2 minutes. If somebody has a question, maybe one question, you can raise your hand and deliver your questions. Anybody? No? This is a very, okay.
Oh, there's one question.
Yeah. Please go ahead, Bu.
Okay, thank you, Dr. Vimal, for the lecture. I would like to continue my questions with regard to the testing. Yes,
Yeah.
if this gene is really still, let's say, maybe not known, or as you mentioned, it's a kind of an accidental gene. But in Indonesia, we do have now some providers or genetic testing providers which offer their services to have a genetic testing. Although most of this genetic testing I use for not in relation with nutrition, but more on the diseases, let's say. But I'm sure there might be, this area would develop more sooner. But then, what do you think about this genetic testing in general? Let's say whether we do need to do that, or how should we choose later if we do have some services? Thank you very much.
So the genetic testing for monogenic diseases is totally acceptable, because in monogenic, it's only one gene. You're just testing a mutation and you know that whether you have the disease or not. So it's more of a genetic disorder. But here we are talking about obesity, diabetes, cardiovascular disease. As I said, like I was talking, these diseases are complex traits. We still do not know what are the factors which are contributing to these diseases. And that's the reason why different public health organizations are providing funding to carry out these research activities. If you already know that all the factors are available, then why would they fund for these research activities focusing on obesity or diabetes or cardiovascular disease? So we are still trying to figure out what exactly is happening with obesity. But of course, there are several gene testing and personalized nutrition centers, not only in Indonesia, all over the world. So I'm totally against all those gene testing centers, because they are providing personalized diets which are working for some people and which are not working for other people. Because what they're trying to do is they are trying to utilize the findings from the Western population, and they're trying to provide the personalized diet for lower middle income countries. So that is exactly the reason why it works for some people and it's not working for some people. And it could also be dangerous to some people who are not able to digest the kind of dietary pattern which is more suitable for the European population. So it can also lead to other inflammatory bowel problems. So I'm totally against the personalized nutrition strategy at the moment, what people are trying to do, because that is not the way, and that is not what I'm thinking as part of precision nutrition. And nobody is using a precision nutrition approach, which is going to be the future, because we do need a lot of artificial intelligence as a support to reach that level of gene testing and precision nutrition.
Thank you very much, bro.
Thank you.
All right, thank you, Professor Vimal, and thank you, Dr. Siti, for your questions. I think we are running out of time, so I need to stop the session, the discussion session for now. I hope everyone have got more insight about the precision nutrition using nutrigenetics and nutrigenomics studies. And as a take home message today, I think like Professor Vimal has mentioned, the genes load the gun, but the environment pulls the trigger. We might have the high genetic risk, but we can control our lifestyle. So with that, would you share your screen? All right, so as as the token of like a symbolic of our appreciation to you, we would like to give you this as the symbolic of token of appreciation. Thank you, Professor Vimal, for sharing your words with us. This is so insightful and hopefully also inspiring for all the attendees who are joining us today. So thank you. We can give some virtual applause perhaps to Professor Vimal.
Thank you very much for your kind message and for the certificate of appreciation. And apologies for extending over time. And I hope you all enjoyed the lecture, and I'll see you again sometime in another occasion.
Yes, thank you, Professor Vimal. I believe we enjoy all the knowledge that you have shared with us today. And I also would like to thank you, all the attendees, for participating in the Power Talk today. Before leaving, please do not forget to fill the evaluation form or survey in order to get your certificate and also to give feedback for us how to improve this Power Talk in the next event. So with this, I would like to end the Power Talk today. See you in another event. And once again, thank you, Professor Vimal, and see you again.
Yeah, thank you, everyone. Bye-bye.
Bye.
Thank you, bro. Bye-bye.
Bye-bye.